All right, guys. So we are going to go ahead and get started. So today we are going to talk about vascular plants. The first group of vascular plants that we are going to talk about is seedless vascular plants. Right. All right. Let's move on. Now, so if you look at this cladogram, right? We have seen this cladogram before. It's the same one that now I'm focusing on this particular group, right? And then last time we talked about this non-vascular plants, right? That was one of the most basal land plants. And then we are looking at these two groups right now. We are looking at actually just two different clades or two different groups. And then in these two particular groups, we have lycophyta and monophyta. So, and we will talk about different compositions, what we find in those particular groups later on. And what I do want to mention right over here is actually this particular point, right? Origin of vascular plants happened about 475 million years ago, and origin of land plants occurred about 475 million years ago. So it took actually about 50 million years for the vascular tissues to evolve in land plants, right? So that it's that. So in other words, the non-vascular plants were the only form of plant species that actually was on Earth for about 50 million years. And then the second part I want to highlight is actually that simply because I group lycophytes and the true ferns, lycophytes are called club mosses, the pteridophytes or monilophytes are called true ferns and relatives, right? I'm teaching both of them today, and I group them as seedless vascular plants, but seedless vascular plant clades are not monophyletic. Right, that is something I want you to understand very well. Simply because I'm grouping them together. I group them together because of convergent evolution reasons, right? They have the same appearances, they have the same life cycles, same attributes, same ecological features. That's why I group them together, right? That does not mean that those two groups are actually related, right? I mean, if you look at the phylogeny, they are not monophyletic, right? They don't have a recent common ancestor, and they are not sister taxa either. Okay, moving on, it's kind of showing the same figure in a different aspect, right? Kind of shows the actual more true diversity. There are multiple groups of non-vascular plants, and then you see the two groups of vascular plants. Understand that there are other vascular plants other than the seedless group we are talking about today, the gymnosperms, right? And then angiosperms, that is the flowering plants and the non-flowering plants that produce seeds are also vascular plants. And then collectively, all of those are parts of the land plant group. And in fact, origin of the seed plants, only happened, about 305 million years ago, right? So again, collectively, the vascular plants that does not produce seeds and the non-vascular plants dominated as a plant life for quite a long time, over nearly about 100 million years. So now the terminology part. Now this is something I should have introduced previously, but I figured that some of you are struggling with the terminology, so I'm explaining the terms now so that it applies to the stuff you learned last week and this week. Micro means either maleness, right, or the male feature, or smaller in size. Mega, larger, right, large size, or female features. Phyl means leaf, right, and phyte means plants. Angium or angia is plural, angium is singular. That refers to vessel, something that either conducts or collects or retains. For example, sporangium means things that actually is a vessel to store spores. Cyte means cells. And sporo refers to spores, right, or something related to spores. Homo means same, like homospore, right? Same spores looking similar. Hetero means different, heterospores, different spores, right? Now, going back to the file and phyte, phyllid means leaf-like structures. Phyte, gametophyte, a plant-like structure that produce gametes. Okay. Then moving on, just to iterate that these are the two phyla that we are that you are going to study right now. Phylum Lycophyta, we do call them like as a common name Lycophytes and phylum Pteridophyta as pteridophytes, but I can also offer you other alternative common names. For Lycophytes, I use the name club mosses, and for pteridophytes, I also use the name ferns and fern-related plants, right? So moving moving on, and do remember always, right? Whenever we talk about a particular species, populate that in your species list in your lab notebook. Now, Lycophytes, I did mention that includes club mosses in general, and there are other types that we also include on there. Spike mosses and quillworts, we will talk about a few examples down the road. And monolithites, also known as pteridophytes. Now, Monilophyta is more of a recent taxonomic name. Pteridophyta is actually a name that we use quite for a while. Both names are still used, interestingly, right? And that includes true ferns and fern-like plants, including whisk ferns and horsetails. And again, we will talk about a few examples later on. All of these groups, lycophytes and pteridophytes have true leaves, stems, and roots. So because of that, they're actually true tissues. In contrast, non-vascular plants had phylids, right, and rhizoids, right? They didn't have any true leaves, right? And the reason why they are not true leaves, just phylids, why they are rhizoids, not roots, is because those structures, phylids and rhizoids, did not have vascular tissue. Didn't have xylem, didn't have phloem, right? So vascular tissue are largely those two, xylem and phloem. And xylem, the job of the xylem is to conduct water and minerals absorbed from the roots and then conduct through the stem into the leaves. Phloem, sort of the opposite direction, where phloem distributes organic food, carbohydrates, that is synthesized in the plant leaves or other reproductive organs to rest of the plant body. Now, I do want to mention something really important here with regard to phloems. I don't know how far you have gone regarding plant histology in your lecture, but phloem also, like when food is stored in roots, the phloem helps to transport those stored food from the roots to the rest of the plant body. Then sporophytes of seedless vascular plants are the larger, more dominant generation. Remember the alternation of generations that we talked about? Now, in the life cycle, in seedless vascular plants, sporophytes become the dominant, right? Gametophyte becomes, less peculiar, less prominent, and smaller, and sporophyte now becomes larger. It's completely opposite when we talked about non-vascular plants, right? And there is an exception to this rule always, guys, like I will talk about that in the exercise. There are always, biology, guys, is not black and white, okay? It's a very gray discipline. It's a gray science, right? Meaning it doesn't mean it's a bad thing. I think it's a great thing. We always have to learn exceptions. Vegetative reproduction via fernlets and bulblets. Now, they can produce sexually and they do produce asexually. So what happens is actually when the front or the leaf of a particular fern touches the ground, it either produces a tiny plant called a fernlet, right, or produces a a bud that can grow as an independent plant. It's really interesting that only happens if the leaf touches the soil, right? It actually stimulates something in the plant leaf, right? Actually it involves hormone synthesis and hormone redistribution and hormone production. And through that, it either creates a completely new plant or directly, or produces a bud that later becomes a plant. And the gametophytes are smaller in size, and they grow on or below the soil surface, right? And yes, so the gametophytes sometimes you may not even see, right, to your naked eye, because because it's extremely small, not microscopic but small, but they largely are underground. That's why. So and let's move on. So why is it evolutionarily beneficial to have vascular tissue? Because as a specialized tissue to conduct transport nutrients, water, and minerals and food, it actually allows the plant to grow larger. It also helps with the water transportation. Now you can actually have very complex tissues, meaning the plant can become larger, bigger, and taller, right? And can grow upright rather than spreading along the ground. So these plants still do not produce seeds. They do not produce flowers. There might be flower-like structures, but they are not true flowers. And then you have the haploid unicellular lightweight spores instead of seeds. Yeah, they still don't produce seeds. Instead, they produce spores. And spores, remember, are haploid, right? And they're extremely lightweight because they can actually spread through the wind, and still depends on water for fertilization. And as the sperm, so the reason is the sperms, they have to actually swim from the male structures, get into the environment, and then find the female structure, and then sort of fertilize the egg inside the female structures. This is actually why ferns and other vascular plants that are seedless are more abundant in wet, damp, moist environments. Now, do not misunderstand. Again, there are exceptions to the rule. As an ecologist, I love talking about exceptions to the rule. There are certain ferns that are well adapted to grow in deserts, right? And I will talk about an example later on. So first, some examples over here that I'm showing to you. Field hostel, whisk fern, those are actually very unique type of seedless vascular plants that actually doesn't have well-developed leaves. The green part you see are actually stems. Then you have the cinnamon fern. It's really interesting. It's a very common plant and it grows around here a lot. And they can grow very tall. Sometimes, like, I should say about 3 feet tall from the ground. And they produce, this very unique reproductive structures, reproductive fronds separate from the non-reproductive frond. And it's called cinnamon fern because the fronds actually have the cinnamon-like bristles growing on it. And holy fern over here, it's called holy fern because the leaves are actually like American holly. And then we have this really cool Japanese painted fern. I just brought it up because it's actually really pretty. You can clearly see the veins of the plant, right? Not the stem. This is actually, this particular part. This particular part is the leaf, right? This is the leaflet. This is the leaflet, right? So, so again, going back over here, this midrib and the other veins are actually colorful, right? And then we have here maidenhair fern, this one, right? So all of those are actually really highly used in ornamental gardening, right? Particularly this Japanese painted fern. These are not, horsetails and whisk ferns are not that popular in ornamental gardens, but most of the other ferns are actually quite popular in the for urban gardening. Sorry, for home gardening, urban gardening, for any kind of ornamental gardening work, right? So these are examples for the phylum Monilophyta, also known as pteridophytes. Now, some examples for Lycophyta, also known as club mosses. Generally, they are known as club mosses because they have this club-like structure that is producing spores, right? One of the most common ones that I'm pretty sure that you guys might have seen are these spike mosses. Again, don't be fooled by the term moss, right? They're actually not a moss. They're not a true moss, right? They're actually a fern, right? Or rather, it's more related to ferns, or more similar to ferns than to mosses, right? And then the quillwort, it's kind of one of the strangest looking plants, right? And you can see they can actually grow in water. They can tolerate complete inundations of water, right? And then it's really agonizing when it advances on its own. And then these dagger leaf club mosses. Now, dagger leaf club mosses, right, are actually epiphytes, though they grow on other ferns. And then, so princess pine is actually another one I want to talk to you about, because princess pine is also fairly common, right? They grow naturally quite a lot in woodlands around here, right? So, like, again, spike mosses, princess mosses, they are not true ferns, right? And princess pine, those are perennial, right? And most ferns are actually perennial, meaning they actually don't repeatedly die every year, right? They remain evergreen, and they actually have very extensive roots. You know, the plant itself is extremely small, right? What, a couple of inches off the ground, right? And sometimes they are known to grow as long as a foot, but mostly they're actually much shorter than that. And this is actually their spore cluster. That is actually where they produce spores. Mostly the stem is actually, the entire plant is about, few inches, couple of inches, 2-3 inches in height. And this particular structure could be about what, a centimeter, a couple of centimeters in height. And in fact, their ancestors, right, about 300,000 years ago in the Paleozoic era, they were known to grow really, really, really tall, right? I'm talking about 30 meters tall, right? So some of the fossil evidence we have of the relatives, closest non-living relatives of princess pine, could be extremely tall. And then about spike mosses, these guys, they are, like I mentioned, they're actually, a very close cousin of the true ferns. They actually grow like a mat around the ground. Although they have vascular tissues, they don't usually grow taller. It's quite interesting. We don't know why they don't, because they actually do better by growing along the substrates, wet substrate in particular. All right, let's move on. Now, again, a few ferns that I wanted to introduce to you, because not to make your life more miserable, but because, it's interesting to learn about plants. Sensitive fern, again, very common around here. You may think sensitive fern as a scrub or a bush because it grows like a scrub or a bush. It's not a scrub or a bush, right? It's a fern that grows like a scrub or a bush. They are called sensitive fern because they are very sensitive to the frost formation. And then we go to this interrupted fern. It's a really cool plant because interrupted fern is called interrupted because in a given frond, right, in a given leaf, you have both photosynthetic non-reproductive vegetative structures, right, as well as reproductive structures on the same frond, right? The same frond or the same leaf contains both non-vegetative and vegetative parts, right? And you will immediately think when you see something like this, oh, some of the leaves have died. In fact, my research students in the field, they tell me, what kind of a disease is that? Why some of the leaves are dead and brown and others are green? Well, it's not a disease. It's actually, these are brown because they are reproductive structures. And in fact, interrupted fern is one cool example for evolutionary stasis. Evolutionary stasis stands for a evolutionary phenomenon where very old plant species remain unchanged for millions of years. In fact, this plant evolved about 180 million years ago, right? And they remain unchanged since the evolution, because we do have their fossil. So we do have plants that are literally fossils that are about 180 million years old. And if you compare those fossil records with this, they haven't changed. And in fact, some amber evidence have enabled us to compare DNA and chromosomal structure. The chromosomal number, the chromosomal structure hasn't changed either. So they are like virtually have undergone evolutionary stasis. No change at all. And do want to talk about these kind of really weird looking ferns, scaly ferns, right? And so the scaly ferns are called scaly because their leaves look like scales, not like leaves, right? They're very thick and succulent in terms of their appearance. And I do also want to talk about this really cool. First, this one, royal fern. Royal fern has, instead of producing, reproductive structures within the same leaf, they produce their reproductive structures separately from the rest of the plant, right? So royal fern and interrupted fern, both of them, as well as sensitive fern, they grow quite tall. In fact, royal fern can grow much taller than sensitive fern, and they also create this scrub bush-like appearance, although they are not actually woody plants at all. I do wanted to talk a little bit about these guys. They're really, really cool. They're actually very rare when they occur. Appalachian bristle fern, they're quite rare. In our state, in Massachusetts, they only occur in the Appalachian area, in the mountains, in the Berkshire Hills. You don't find them around our area in the eastern part. They are extremely diverse and relatively commoner in the southeastern part of the country. The cool stuff about this, they grow in extremely wet and humid places, particularly in like rocky surfaces, wet, humid rocky surfaces, particularly around waterfalls. There's plenty of rock bathed in water. Inside caves, as long as they're exposed to some sunlight, or rather on the edges of the caves were exposed to sunlight. And on caves, they actually grow in these crevices, right? So you might not even see them, and they're extremely small in terms of size. Now, in ferns, generally, I mentioned, right, whatever that is physically observable, the larger part, the most dominant part, I said generally for vascular seedless plants is the sporophyte. But this is not the sporophyte. This is the gametophyte that you see, right? So they actually completely reversed their norm, right? And certain species of Appalachian bristle ferns, or the certain species of bristle ferns, do not actually produce a sporophyte at all. That's one species to do that. Certain species only produce sporophytes once in a while, right? Particularly if they want to reproduce sexually. They produce a strange spore-like structures on the gametophyte. If you look at the gametophyte, and if you look at closely, you see these kind of tiny, tiny, tiny things, right, all over the leaf, all over the leaf tips. And if you're closer, those leaf tips looks like this, right? They can produce spore-like structures over here, right? But the interesting part is that kind of spores that is produced from the gametophyte tend to be like a clone. They propagate clones. But if those spores are produced by actual sporophyte, those spores actually can result in genetic diversity. So it's really cool. Ferns are really cool. They actually define all the odds that you can think about. Is there anything else I want to mention here? Let me think for a quick hot second. Nope, that's good enough. So talking about their histology, right, tissue organization. So they have true tissues, right? They have stems, leaves, and roots. So this is actually the actual leaf, right? The leaf blade. So this part is called the leaf blade. And this area where you don't have any leaflets are called the stalk or the petiole. The entire part is called the frond. Better yet, leaf or sterile frond, right? Or, well, I'll come to the sterile frond part later on, just frond for now, right? And then the tip of that is called the apex, right? And the middle part is called the midrib, right? And these are called either leaflets or pinnae, right? And you can see in certain ones, the pinnae also branches again, and we call them pinnules, right? Then the subterranean, the underground part, right, is actually, this is the rhizome. Now, in fact, you might ask me, okay, so is this petiole the trunk? No, that is part of the frond. That's part of the leaf. No, that's not the trunk. The actual trunk is this, the rhizome. The actual stem is the rhizome, right? And the roots comes out of the rhizome, right? So the rhizome or the stem is actually horizontal, not vertical. It's really interesting. The actual upright part is the leaf. Moving on, the leaf sizes vary a lot. Arrangement and the shape varies a lot. And some examples right over here, right? So, sometimes you actually have this simple single leaf fronds, and sometimes the leaf frond divides into leaflets, right, or pinnae, and sometimes divided into pinnules, right? So it could be extremely complicated leaf blade structures, right? And sometimes the leaf could be like scales, right? Or extremely small, like in this Selanginella. And also the location of the spore producing structure, the sporangia, can also vary, right? I will show you examples over here, right? So in certain cases, the sporangia occurs on the underside of the leaf frond, right? Like in this case, sometimes on the tips of the leaf frond, but on the underside. And in certain cases, the sporangia can be produced separately, right, as a separate fertile frond, right? So if the sporangia are produced on the underneath of the frond, right, like here or like here or like here, we call them sori. Each of these things is a sori, right? I will show you a blow up image later on. And sometimes it actually is produced as a strobili on the top, on the tip of the plant, like in the case of princess or club ferns or club mosses or princess mosses. I will show you those images again separately. Presence of the vascular tissues allow us to now use the term leaves, stems, and roots, right? From this point onwards, we can actually use those because those are true tissues, right? They have vascular structures. Now, know yourself that what cells makes up the vascular tissue. You will cover those in lectures. And when talking about the leaves, we can use modifiers such as like large leaves, macro, small leaves, micro, microphyll, small leaf, megaphyll, large leaf, right? So we can also talk about how the leaves are arranged. Are they arranged like a flower, whorled, right? Where you have multiple leaves occurring around a particular area around the stem, right? So all of those differences can be used to characterize a particular species and differentiate between different species, or to describe different tissues, different components of the plant body. We can talk about locations where the organs are found as well. Node, meaning actually the particular part of the stem where leaves are attached, right? Internode is actually the space between nodes, right? Now, we actually just mentioned this part. Now, this is an example for the strobilus, with respect to princess moss, also known as princess fern, right? Where this is the strobilus, this is the collection of sporangia, right? And over here you are looking at the fertile frond produced separately, right? And sori underneath the leaf blade. Now, if you look at a strobilus like this one, right, and look at it under the microscope, you will actually notice two types of spores, right? Microspores, in this case, those are microscopic, very small spores, right? They are produced in microsporangium, right? So Strobilus contains multiple sporangia, and some sporangia are microsporangium producing microspores, right? And this case, micro is actually referring to something smaller and also refers to maleness. Those are male structures. Microspores, when they germinate, they produce male plants, right? And or male gametophytes. And then we have megaspores. Look at that. Those are very large spores, right? So they are mega, meaning they are large spores produced in megasporangium. And mega also refers to the fact that they are female structures. So those megaspores, when they germinate, they produce female plants, female gametophytes. The leaf that occurs around the megasporangium is called megasporophyll or megaphyll. The leaf that you find around microspore is known as microspell. I mistyped over here. It should be microsporophyll. I will actually correct that and upload it again, right? It should be microsporophyll or microphyll, all right? Do make that correction on your notes. This is not mega, this is micro. So here, this is the first time seeing the production of two different types of spores, right? That we can physically see as different. So because of this, we can tell a female from a male spore, right? Just by observing them under a microscope. The production of the two different types of spores is called heterospory. Hetero means different. Spores are spore. Spory refers to spores. Selaginella, right, is the only group that you need to worry about with respect to heterospory. All the other ferns, right, all the other seedless vascular plants are homoporous, producing the same kind of spores, no male or female spores. Selaginella is the only group of seedless vascular plants that you need to worry about, right, that are heterosporous, right? So sporangium, if you actually look at a single sporangium, right, of other ferns from pteridophytes, if you take a close-up of a single sporangium of a true fern under a sorus, remember what a sorus is, right? Actually, these structures are actually considered called sorus. If you look under one of those, it looks like this, right? This is a single sporangium, and they occur in clusters, and a single cluster of sporangium is known as a spore, as a sori or sorus. I will show you a diagram later on in the exercise. Its job, what does it do? Like, you look at the components of that. It has spores inside, and then surrounding the spores is actually something called annulus. Annulus over here and source inside, right? And the annulus, as you can see in both diagrams, their cell wall thickness is different. On one side, the cell walls are thicker. On the other side, the cell walls are thinner, right? So because of the differential thickness, when they absorb water or when they actually get dehydrated, they expand and contract differently. That differential expansion and contraction helps to actually create rupturing, right? Particularly in drier environments. In wetter environments, they actually absorb water, and that actually creates a stronger integrity. But if in dry environments, they actually dehydrate, and the dehydration is extremely important to crack the sporangium and create lips, right? And create this lip-like opening. And through the lip-like opening, the spores actually can exit the sporangium and spread through the wind, right? Okay. Last part in this introductory video, before we go to the interactive part, we'll talk about the sperms of the fern. Now, I actually have a couple of really great videos to show you the actual mobility of the sperms. We call them sperms because they actually move, right? When they are first released, they look like this, right? You can see some of them actually exiting, right, the female reproductive structures, right? And when these sperms microscopically looks like this, and they appear sort of, jittering over here, not because it's a bad photograph, because they are actually moving, right? So when they actually move so fast, it's hard to capture a good photograph. That's why they appear sort of wiggly over here, right? Because they actually do wiggle. If you take a close-up photograph, they actually, so close. This is the female reproductive unit that sperms comes from. Sorry, the male reproductive units that the sperms actually come from, archegonium, right? The actual sperms looks like this, right? Again, you can see they are not, easily photographable because they move so much. And then if you actually take a few and, look under a very good microscope, you can actually see how, that they have so many flagella around a single sperm. This is the actual sperm head, right? This is the pink color is the actual sperm. The light pink parts around them are actually the flagellum of the sperm, right? So this is actually an electron microscopic photograph, right? The actual sperm head, and then all over it, the spaghetti-like parts are actually the flagella of the sperm that helps them to swim, right? And in fact, the number of sperm, the flagella, right, is variable, right, around different species. So that actually helps us to understand or identify the species. So in fact, if you just look at the sperm without looking into the genetics, you can ID at least the genus, maybe not to the species level, but at least to the genus level. And do not forget that bryophytes also have sperms. They produce sperms and they can also move. So they too have flagella, right? Algae also produce sperms. They too have flagella. So having flagella is not a fern character, right? But what you do see is actually that ferns have a greater number of flagella per each sperm, but bryophytes and algae do not, right? And some algae only has one flagella or two flagella at most, right? And bryophyte could have a fewer more, two, three, but some early vascular plants, right, ferns could have actually, a lot. I mean, look at over here, I can barely count. All right. Okay, that's it about the introductory part. Any questions before we navigate to the interactive part? Any questions? So now I actually, yeah, go ahead, ask.
You said the males come from the Archegonia?
Yeah, I do have to correct myself, Emily. You're right. Males comes from Archegonia, right? And sorry, Antheridia. My bad. Antheridia. Not Archegonia. I do have to correct myself. The photograph was misleading, so that's why I make the mistake. Let me go back. Thank you for asking that back. Actually, my yeah, over here, like actually the reason why the photograph is over there, it keeps confusing me over and over, because the sperms are trying to fertilize the egg inside the archegonium. So the sperms are coming from Antheridia or Antheridium, right? Here, what you're looking at is sperms being released from the Antheridia, right? And now over here, you're looking at sperms swimming into the archegonium. Thanks a lot for clarifying that. It always mixes me up. When I prepared the PowerPoint, it actually confused me, but I get the point. It's actually sperm swimming towards the archegonium. I actually read the exact paper to understand why they put it. Actually, it makes sense because sperms are swimming to archegonium to fertilize the egg inside the archegonium, right? Any other questions? So just to show you, I mean, this is actually, come on. This is, you can see sperms moving in this particular video. And you can actually enjoy it yourself. So they actually move, quite fast and quite robustly, right? Okie dokie. Now let me actually quit sharing that and move to the new lecture through the exercise. Any questions, anyone? All right, guys, now we are going into the interactive part where we are going to ask questions and questions. So please do open your cameras, and if you do, I would really appreciate it. It makes things extremely comfortable for me to carry on the conversations with you guys. Okay, let's go on this lecture now. So, let me do this. Good. Let me go to the beginning of this. Right. So, first job that we are going to do is to go through the dichotomous key. How do we do the dichotomous key? Well, first question to you guys. I know most of you are getting familiar with this. Shall we do one example or shall we just skip this part if it is easier? One example, Emily? Okay, very well. Let's do one example. First part, read with me. 1A, plant erect. Okay, let's look at the plant first. This is the first plant, right? And what do we see here? This plant has frond.
You're not sharing the PowerPoint.
Oh, damn, thank you. See, like, you know why I hate online teaching? So yeah, now we do, right, Emily? All right, do we see it now, just to make sure? Yes, very cool.
Yes.
So this is the plant that we are looking at. Look at the PowerPoint. Yeah, and it says the plant has fronds and leaflets, and this plant, this is a leaflet, like I mentioned, right? And this whole part is the leaf or the frond. Small portion, it is a small portion of the larger leaf. Let's go back to the key. The plant erect with needle-like leaves, scale-like leaves, or leaves absent, right? And the B, plant with fronds, long, wide leaves with stem-like veins and leaflets. Where do we go? 1A or 1B, right, guys? This is the interactive part. Go to 2 or go to 5? Let's look at the plant.
Go to 5.
Go to 5. Yes, that is correct. Go to 5. Because what is needle-like? Let me show you what is needle-like. That is can be considered like tiny, right? This can be considered leafless, right? This is fairly needle type, I would say. This is scale-like, right? Yeah, and we give you a description on the top. So yeah, we are going to go to 5, right? And 5 over here, it says leaves composed of many leaflets and closely arranged along a rachis, stem-like vein, or in a single leaf, or as a single leaf, right? And are not bifurcated, not split into 2, right? The B part is single leaf with smooth margins, tips bifurcated, right? Now, let's look over here, right? A, right? I mean, it's not a single leaf, right? And clearly the tips are not bifurcated, right? The tips are actually occurring in a single leaf, right? So we, in this case, move to 6, right? Leaves with many leaflets, leaves simple or lobed. Well, the leaves are not simple, right? Leaves has many leaflets, so we go to 7, right? And 7, it says leaflets tightly arranged along a stem, L-shaped. Leaves with spaces in between, leaflets with spaces in between, holly-like leaves, and not L-shaped. Well, let's look at over here. They are certainly not L-shaped. They're like a teardrop, right? And there's space in between. I mean, remember, this is the, this is one leaflet, this is the other leaflet. There's space in between, right? And let me show you what is L-shaped. This is actually L-shaped, right? And you can also see the leaflets are very tightly arranged on the leaf, right? Now, I do want to mention this, the common name for this one, because A is very common around here. Now, look at this L shape. What does this remind you of? Right? A stocking, right? Particularly a Christmas stocking, right? The one you hang in your fireplace for Santa to, drop gifts. So this is actually called Christmas fern, right? I love fern, so I cannot stop, talking about them. Okay, we will move on. So, and then we will go over here. So because of that, it actually goes to this one, right? It is this particular species H. Okay, the rest you can actually do on your own. Um, I will talk a lot about these kind of plants later down the road. So fern life cycle. So these are the parts of the fern life cycle that I want you to know specifically through diagrams, right? Now, a question for you guys. I need a shout or a nod, for those who are on the video with me. How many of you are still struggling to understand diploid, haploid that want me to explain? How many of you are struggling with mitosis and meiosis? Yeah, I actually see people saying no, they're fine, right? Yeah, okay, very good. So last time, give me a shout. If I should go over this part really quickly, but if not, I'm skipping. Seems like you're telling me to skip, which actually doesn't surprise me. Yeah, skip. Very good. If you're struggling, if you're shy to speak, ask for the after the lecture, or you can read it yourself. That particular slide is so self-explanatory. Okay, let's look at the life cycle now over here, right? Let's start with the numbered one over here. What I'm looking at is a spore, right? So the most ferns are homosporous. All spores looks like the same, and they actually are released from the sporangia, and they land on the soil and they start generating, right, or germinating. The fern spore develops into a small heart-shaped gametophyte. Now, those gametophytes, right, some of them could be, at least during their early stages, they are photosynthetic, right? They can photosynthesize, right? But during the later stages, when they grow larger, when they particularly go underground, they might not be able to photosynthesize. Each gametophyte has both male and female sex organs. Like you see, the same plant gametophyte contains both antheridia and archegonia, right? But the archegonia and antheridia usually mature at different times. The reason why those two organs mature at different times is to prevent self-fertilization and to encourage cross-fertilization. And then you can see a close-up over here, the archegonium, the female part, and the male part, the antheridia, right? So antheridia produce sperms. Sperms swim and enters the archegonia. The egg remains inside the archegonia, right? And the fern sperm, like those of all seedless vascular plants, use flagella that beats around to swim through moisture, through water, right? Actually, it's not just moisture, not just humidity. No, they actually require standing water, right? A thin film of water, maybe, but they need that. And that goes into the antheridia. Sperm go into the antheridia, and the egg inside the antheridia become fertilized. Egg inside the archegonia become fertilized. And then after fertilization, the egg becomes the zygote, right? Remember the ploidity. Sperms are single N, right? N, right? Haploid, right? Gametophyte, haploid. Egg, haploid. Sperm, haploid. Zygote, diploid. And then that zygote undergo mitosis, right? And after mitosis, you actually start development of the sporophyte. Sporophyte now actually emerges from the gametophyte. At this stage, gametophyte starts to wither, right, and deteriorate or become underground. And you can also see the gametophyte actually produces not roots, rhizoids over here, right? And these rhizoids still remain there because it will require to absorb minerals and nutrient and anchor to a place, right? And provide nourishment for the developing sporophyte. And then in the sporophyte, it produces a single set of leaves first, and then the other leaf, the first leaf is like this, the second leaf looks like that, right? And then the first leaf later develops into something like this, right? So the young sporophyte looks like this, but more developed sporophyte look like that. Let me actually move this to somewhere else so I can read it. So over here, fertilized egg develops into a new sporophyte and the young plant grows out from the archegonium of its parent, the gametophyte. Remember, if I ask you, where do you find the young early developing sporophyte? It's actually developing from the archegonium, right? And so it is still located on this gametophyte. And then over here, this you have much more well-developed mature sporophyte. When a sporophyte matures, it actually has sporangium on them, right? And then, of course, spores undergo meiosis and produce haploid spores, and the life cycle continues. Now, the part I want to explain is right over here, right? No, right? This, right? Again, so you're looking at a single front, and this is the blade part. If you look at the leaflets on the blade, you see, like in each pinnae of the leaflet, you see those sori. Sori is a collection of sporangium, right? And then again, what happens is as the sporophyte matures, the actual stem, the rhizome, keeps growing horizontally, and it will keep producing different leaves, right? So youngest leaf looks like this. So it's only the very first leaf that looks like that. The second leaf looks like this. It sort of comes up as a coil, right? And then it grows into something like that, right? And then in fact, the rhizome can keep growing like further apart, not produce any leaves, and produce an entire fern on a further away from the first set of ferns. So when these ferns grow, you don't see usually a single plant, you see a cluster of plants, right, of different ages. I've seen once in a while like cinnamon ferns and interrupted ferns growing singly, but they won't remain singly for that long, right? So if I zoom into a single pinnae or a leaflet, it looks like this, right? So each structure is called a sorus. This thing is called a sorus, and what you see inside of a sorus, right? The sorus is actually protected by a structure called indusium, right? And if you zoom into the edge of the indusium, you actually see sporangia, right? So first, the indusium actually protects the actual sporangia inside it, right? But later, as the sporangia matures and they grow larger, they sort of protrudes out of the indusium and kind of protrudes out like this, right? Okay. Not going to go over this, but I do encourage you to sort of study it yourself and also watch this narrated video that actually very nicely describes the entire life cycle, right? So let's talk a little bit about the fern gametophyte because it's very inconspicuous. The job of a spore is actually to create this gametophyte, right? And most ferns are homoporous, so each spore produces a gametophyte that has both male and female reproductive structures. Male reproductive structure and 3D female reproductive structure, archegonia, right? Producing eggs, and 3D producing sperms. And the and this in contrast to mosses that actually have separate male and female gametophytes, because if you remember, in mosses, right, they actually have more dominant gametophytes, right, and more less dominant sporophytes. But the things are quite different in the case of ferns, right? So once a spore has germinated, it produces a heart-shaped gametophyte, and that heart-shaped gametophyte of ferns have a new name, protothallus, right? Now, is protothallus a part of the gametophyte? No, it is not. It's not a part of the gametophyte. The gametophyte of a fern has a name, protothallus, right? So it's kind of another name for it. And again, the previous point, in mosses, you should also remember, in the bryophytes, you should also remember that they produce separate male and female gametophytes, but we don't have separate male and female gametophytes in ferns. It's actually the same gametophyte producing both male and female reproductive structures. Keep that in your mind. The second part I want to iterate is now the gametophyte in ferns, most ferns, is not dominant, is smaller, tinier. Sporophyte is actually larger, more dominant. Now, protothallus of the fern is sort of a reminiscent of the thallus of the liverworts and the hornworts, right? So that's actually something you can make the connection to. When evolution happens, they don't lose their differences. They actually evolve to a better, evolution happens towards better adaptations, right? So, but some of the very primitive characteristic or the basal characteristics does not completely go away. So the thallus-like gametophyte shape is still remains, right? That's why the gametophyte of the fern is still looking like a thallus, right? And hence we call it prothallus or protothallus. In certain literature, I have read the word protothallus, and protothallus is actually another term that we use. The same thing, but I don't care what we, what you use. But I correction, I do care what you use because I do want you to know what we call them. I kind of hate this advancing on itself. Let me stop it from advancing itself. Yes. Yeah, know the terms, right? So protothallus or prothallus, right? Know both terms because we might give you one or the other in the exam. And once the egg in any of these archegonia has been fertilized, creating a zygote, the remaining archegonia and eggs will abort. So remember, in a given prothallus, in a given gametophyte, right, there are going to be multiple archegonia, right? And each archegonia with a single egg, right? So when one gets fertilized, that particular gametophyte cannot sustain multiple developing zygotes, right? So because of that, they actually, automatically all the other archegonia and eggs inside the archegonia becomes aborted, right? They will cease to undergo any fertilization, right? So the first come first serve. Whenever the first egg that gets fertilized will develop and the others will not develop. All right. So and then again, here I'm providing the example, the words, the terminology that I expect you to learn, right? In the fern life cycle, right? Unlike in non-vascular plants, the gametophyte is no longer the dominant one, right? The sporophyte becomes the dominant. Also, the two generations are now independent. Remember, in non-vascular plants, right, the sporophyte could not photosynthesize. Sporophyte was a parasitic plant, parasitic life history stage on the gametophyte. But now it's not. Both generations can photosynthesize and can be independent of each other, right? Yeah, it is true that early developing sporophyte requires some dependency on the gametophyte, but after the early stage, the sporophyte can photosynthesize itself. This is the first sign that we see differentiation of spores, right? Selaginella was the example where we saw micro spores or the male spores and the megaspores, the female spores. And we can look at the spores and then, physically examine them and tell you whether what is male or female. And that is called heterospory. We explained that already. Now mosses, in contrast, right? All the non-vascular plants, in contrast, are homosporous, right? Meaning that the male and female spores don't look different, right? Now, among ferns, among non-seed producing vascular plants, right, it's Selaginella is the only group that are heterosporous. The rest are homosporous. Now, this is actually your labeling exercise, right? Here, now talk with me. The generation, come on. Generation responsible for producing gametes, right, is what? Gametophyte, okay? It's always, right? Gametophyte, right? And then we ask you, the young plant that will eventually be responsible for producing spores, the one that is sort of sprouting up over here, right? What is that is called?
Sporophyte.
Correct, that is a sporophyte, the structure that holds the fertilized egg. Be very careful, structure that holds the fertilized egg, right? What is that?
Is that the Archegonia?
Correct. Very good, Matt. That is the archegonia, the female reproductive structure that contains the egg, right? Now, I'm telling you guys, if you very well understand the life cycle, right? If you remember that very well, if you understand that very well, the rest of the thing I'm teaching is a piece of cake, right? Now, um, I mentioned that the gametophyte that blown up over here is extremely small, right? So in the real life, that it's hard to find them in the nature, right? This is actually a few photographs that I was able to find, right? Where you see over here, where you see the sporophyte, right? The sporophyte coming out of the protothallium, right? Or the prothallium, right? This is the prothallium part, and this part is actually the erected part is actually the sporophyte. This is the prothallium or the gametophyte, right? Or the protothallium. Now, over here, actually, we are labeling it. This is the prothallium or the prothallus, right? And I know there are several ways to pronounce it, but I use the word that I use consistently. I consistently use prothallium, right? Prothallus is nothing different, another very similar name, right? And but remember, right, in your terminology list in the lab notebook, if you enter all of these, as with the same definition in the exam, you should not get confused. You can see the young sporophyte upright, young sporophyte upright, young sporophyte emerging upright, emerging from the prothallus, right? Look at this, emerging from the prothallus or emerging from the prothallium, right? So yeah, so in nature you can find it. You have to work really hard to find it, right? And I worked really hard to find these examples. I do a lot of field work, so it's not that easy to find these examples. But kind of fun to see them, in nature. So okay, over here, so here you can see the actual, like much early sporophyte, right? Growing from the archegonium. The browner part you see over here is actually, the archegonium, right? And this is the prothallus, right? This is the prothallus, also known as the gametophyte. And here, right? So early it starts like this, and later when it germinates, it looks like this, right? So now notice this in the prothallium, right? These tiny hair-like structures that are coming out, these tiny hair-like structures that you see over here, those are rhizoids. Those belongs to actually the gametophyte, the prothallium, right? But when you see, the sporophyte at least, this large, but it is, when you see the leaf emerging like this, the first leaf emerging like that, and the shoot very well developed, you will also see the true root emerging, right? And from the root, you can also see true, small miniature hair emerging as well. So, and now, again, I did show the previous image, but I'm showing this again. It's extremely important to understand that the root, the actual stem is underground, the rhizome, right? And the actual root comes right over here, okay? Yeah, I did describe this part before, so I'm not going over this again. Now here, again, it's your job to label them, right? I just labeled something very similar, so I'm not going to go over it again because over here, I went over what those parts are. Oh, one more thing I want to point out is actually, so if I circle this particular one and ask what that is, you are going to call it the leaf or the first leaf, right? Right. This part, the actual, the line that I'm pointing at, those are actually the vascular tissues, right? Those are the actual vascular tissues that is responsible for transporting water, nutrient, and food, okay? So that you can call it the vascular tissue. Right now, I cannot tell you whether we are looking at phloem or xylem. You have to look under a microscope to tell that. But as of now, you can call that the vascular tissue, right? Anything that I didn't point out before that I should? Now, be very careful when you read this. Structure for anchoring. So it could be two things, rhizoids or roots. Structure for anchoring the generation responsible for producing spores. So I'm talking about particularly for those that is producing spores. So your answer is roots, right? And then structure for anchoring the generation that is responsible for producing gametes. So then it's going to be rhizoid. Be very careful what we are pointing at. Okay. Now remember that ferns have stomata. So if you have stomata, right, stomata openings can be controlled. How? Because it has guard cells, right? So the actual opening that I'm covering up with my pointer is the stomatal opening. The bean-shaped cells around here are actually the guard cells, right? That actually protects, that actually controls the aperture of the stomata, okay? How stomatal opening change, how the stomata can be opened fully, partly closed or fully closed, that depends on the contraction and expansion of the guard cells, but I'm not going to go into detail how to right now. I believe your lecture professor might cover it, but I think that kind of detail comes way later in plant physiology. But if you're interested, I can go over that, but it's not, I don't make it imperative at this particular stage. So couple of examples for potted fern plants. So the roots, obviously, you cannot see because they're actually earthened. But when you are drawing, what is more important is drawing the above ground biomass, and particularly where you find the actual sori, right? Again, sori, singular, sorus, plural. Those are the structures that are aggregates of sporangia. Now, looking into microscopic images of sori, right? So we are looking at the entire leaf. Leaf contains multiple sori, and this is a single sorus, right? In a single sorus, you have immature sporangium, right? And sometimes this mature sporangium can detach from the sorus, right? And then subsequently rupture. If actually you take a look at a leaf, right, this is how it looks as a cross section, right? So this is the indusium, right? Indusium looks like a mushroom, like upside down, right? Or an umbrella that is upside down, right? So between the actual leaf and the indusium, you have the sporangia, right? And again, if you look at a single sporangium, it looks like that. Remember, you have the annulus with lip cells around it, and then the spores inside. Remember, it is actually the differential thickening of the cell walls in the annulus that helps for the lip cells to rupture and form that lip opening to help dispersal of the spores. Now, spores, how do they disperse? Not through water, it's through air. In fact, for many species of ferns, the spore dispersal depends on wind and dryness. Okay, so again, now I have labeled this so many times, you should be able to answer these questions on your own. Now, doing that on your own is extremely important. And then a video that is actually really cool, it shows how those sporangia sort of explode and discharge spores, right? Here we are looking at the prothalium, right? And in the prothalium, actually, we are looking at over here. This should actually have undergone. Yeah, here. So this is actually a fern gametophyte, the prothalium, and then you are looking at the two different reproductive structures, antheridia, right, and the archegonia. So, so I'm calling this antheridia because there are two antheridia over there, right, and a single archegonium. They are structurally different in the microscope, right? And in fact, shape-wise, antheridia usually are round and have many sperms developing inside it. Archegonia, on the other hand, they are more elongated, right? Sort of tube-like, and they only have a single egg inside of them, right? Now, again, a close-up of antheridia looks like that, right? And close-up of archegonia looks like that. Again, this is the tube-like formation, right? Now, the interesting part is if you look at that from the top, right? Imagine looking at this from the structure from above, right? It will still look rounder, right? So over here, so this is actually, you have round structure, right? It says produce what reproductive cells by what cellular process. So again, now answer me, right? What what reproductive cells would you expect to come out of this round structure? And it is producing, and those reproductive cells are what? Produced by what process? Answer.
It's the antheridia producing sperm cells through meiosis.
Excellent. Excellent. Is it meiosis or mitosis? Right? Gametophyte is haploid.
So
Sperms are also,
mitosis.
yeah, mitosis. Okay, that's what I mentioned. Understand the life cycle, and then the rest of the part is easy. So it is not meiosis, it's mitosis. Okay. Meiosis happens when you're producing spores. Meiosis happens when you're producing spores. Okay. Yeah. You might be mixing it up because you are thinking about male, like animal sperms, right? Yeah, it's quite different. Now, and then now this is, right, the opposite, right? It's a female reproductive structure, right? Producing eggs, right? Now, these, right? This, that thing, right? These are also, right? All of these are also archegonia producing eggs, right? The reason why they look round is because you are looking from above, not from the side, right? Why I am confident that they are archegonia, right, is actually I don't see sperms inside it. That's actually how I know, right? In Antheridia, you can actually clearly see sperms inside it. Okay. The rest of the part you can actually fill on yourself. Exercise 3. We'll talk a little bit more about the lycophytes, and over here we are talking about lycopodium, right? And looking at a sporophyte, right? So now work with me. Let's start from the bottom, right? And again, please do answer. Part of the plant that is responsible for anchoring is
The root.
root, right? Now that is root. It's not rhizome, right? Because this is not a pteridophyte. This is a lycophyte, right? And this actual part is the stem, right? And then this is a leaf. The tiny needle-like part is actually leaflets, right? And then what is this?
Sporophyte.
Correct. This is sporophyte. This is sporophyte. The entire thing is sporophyte. This?
Gametophyte.
No. Gametophyte is actually extremely small. Sporangia. This is actually the collection of sporangia, right? A collection of sporangia that is known as strobilus. Strobilus, right? And the strobilus in lycophytes are always on the top of the plant. right? It looks like a club. That's why we call collectively all of them club mosses. All right, so this is actually again the same here. Right. And so here we are looking at, we did actually go over this part earlier. We described, in Selanginella, right? In this particular plant, right? If you take a look at a close up on their strobilus, right? You see something like this, and that actually contains megasporangium and microsporangium, right? And in fact, the color of the mega and microsporangium are also slightly different. And if you look at an actual cross section, then you clearly see the differences, right? You do see megasporangium to contain large megaspores. You do see microsporangium to contain smaller microspores, right? Okay, I did explain this part earlier, so I'm not going to spend too much time. Again, just to understand the perspective, this is the spore. Seriously, this is the entire sporophyte. This is the strobilus. And it's actually this strobilus that you look under microscope, which looks like this under a microscope, and that blown up a little bit more looks like this, right? And this is actually sort of a blow up of this, right? Okie dokie. The last thing that I do want to mention is actually, do not forget that these things have names, right? Megasporophile versus microsporophile, right? The leaf that associated with the sporangium has a name, right? So you do need to know that, right? This is actually regarding labeling these things that I just described with you, right? I do believe, I've gone over this 100 times, so I will not go over. But at the end of the lecture, if you do need me to go over, please do let me know. Whisk fern, this is a really interesting plant that I do need you to know about in detail. So these plants, these ferns, the whisk ferns, they do not actually have leaves. So because of that, we don't consider whisk ferns as true ferns, right? So those plants, they don't have leaves at all. They are still vascular plants, by the way, right? You might argue, wait a minute, didn't you tell us that all vascular plants have true tissues and you're telling me otherwise? Well, yes, they actually lack plant tissues. They lack true leaves, right? The reason is actually not because they are primitive, because they actually have developed evolved adaptations to get rid of leaves and do photosynthesis otherwise. So loss of leaves is a secondary evolutionary characteristic. It's a secondary loss, which we called reversal evolution, right? Not a primitive character. Okay. But they have small, instead of actual leaves, they have small protrusions called enations, right? Now, enations are, in other words, they are tiny, tiny leaves. This is how a plant looks like, right? And if you take a closer look at these branches, that the stem itself, the stem branches profoundly, can you see these tiny, tiny spines all over the branch that I'm pointing at? This. All of those spine-like parts, those are enations. Those are actually modified, highly modified, tiny, tiny leaves, right? Sporangia, where are sporangia? Well, sporangia, can you see these tiny yellow bulbs all over the plant? Those are, those balls are actually the sporangia, right? And so I know you are going to hate plant biologists. They give all the different names or the same name for 100 different names for the same structure. So inside a single bulb, you actually have multiple sporangia. So big, and those multiple sporangia actually fuse together, and they actually give a new name for those fused sporangia that actually looks like a ball externally, yellow ball externally, right? This particular structure is called synangium, and inside the synangium, you could see about 3 sporangia, right? Here you're only looking at 2 in my diagram because it's a cross section. The third one you cannot see in a cross section, right? And then actually the enation, the tiny, tiny leaf, could be found immediately underneath the synangia, right? Now, I did not include synangia or synangium as a term that you want to that I expect you to learn, but I put it out there because people use it. Plant biologists use it a lot. That's why it is. That's why I mentioned that over there. Okie dokie. And so these groups, collectively whisk ferns are called silotales, right? With this, the P is silent, so it is silotum, right? The P is silent, and they are the least complex form of terrestrial vascular plants, actually. And they were once believed to be actually very primitive or remnant plants of an extinct Denovian flora. In other words, we actually thought they are also undergoing evolutionary stasis, right? But recent molecular biological analysis revealed it is not the case, right? So this is primarily because the xylophytes are actually the only living vascular plant that do not have true leaves or true roots, right? So they actually still they have been considered primitive for quite a long time, and some plant biologists still consider that, but it is actually incorrect. It's not primitive. So see, certain studies, very recent studies in development, that in the plant development and molecular biology, suggest that the group may be actually a reduced or a highly modified form of fern-like ancestors. So because of that, we now, without reasonable doubt, we consider them as fern allies, right? Or as a seedless vascular plant that is closely related to true ferns. What else? There's a lot that I can tell you about these plants, but I don't want to add confusion right now. If you look closely at that particular plant, you do notice that the stems are green. Now think about why the stems are green, because if they don't have leaves, if the leaves are tiny, they're going to be a problem with reproduction, problem with photosynthesis. So they actually overcome that by adding chlorophyll, right, into the stems. So stems are green because stems have chlorophyll, right? Anything that there's a lot of things that I can talk about these plants, but I don't think it's really necessary at this particular stage. You know enough now. The last thing I do want to mention is that instead of roots, they have rhizoids, and the stems are actually branching dichotomously, right? They branch into two branches repeatedly. Okay, the next group is this, right? Hostels. Now, hostels, these plants have highly reduced leaves. Now, previously I mentioned, I actually said they don't have true leaves, but they have is enations, which is something like an extremely tiny leaf, right? Here they actually do have leaves, but those leaves are reduced, heavily reduced. And so the actual leaves are like this, they look like needles, sort of resembles a tinier version of pines, right? And the plants are highly reduced. They have highly reduced leaves, and they keep branching repeatedly. And the leaves actually sort of, the branches make a wall around the stem. If you can see over here, right? You see a wall, right, of branches around a particular node of the stem. And so they have a true stem, a main stem, and they actually also have true roots. Look at my diagram. They do have, the rhizome, and then from the rhizome, it produces true roots. Now, the interesting part is the spore production, the spore producing fronds are separate from the vegetative fronds. These are the photosynthetic fronds with the leaves, and these are the reproductive fronds that actually contain the strobilus, right? Strobilus has multiple sporangia inside of them. Now, there are leaves on the fertile fronds, right? Fertile fronds are the fronds that actually has the strobilus. Are actually like surrounding each node, you see this extremely tiny leaves. This is a close-up that you see over here, right? This actually looks like sort of teeth, right, surrounding the stem, right, surrounding the fertile frond, right? So those are the actual, those are also leaves, right? Those leaves are much smaller than the leaves that you see in actual sterile fronds, right? And to the naked eye, those plants looks like that, right? And if you look closer in those plants, you will see the actual fertile front looking like this. And here you can see those tiny leaves, right? Surrounding the nodes just like that, right? So you can sort of say that they have two types of leaves. Both type of leaves are extremely reduced. The leaves on sterile fronds are a little bit larger, more pronounced, right? Still needle-like, but if you look at the fertile frond, those leaves are much, much more smaller, right? And the next exercise is actually, I'm not going to cover them right now because one is regarding cuticle. We already covered the cuticle part before, right? And then the stomata part, I covered that in the intro lecture. So look, so you can resort to that, look at the stomata component. So this is really important, right? First, I want you to pay attention to this one. This is actually kind of you are looking at a cross section, right? And the cross section is actually taking, like if you have the stem or the root like this, right? Cutting it across, right? Not lengthwise, right? Sort of the widthwise is actually what you get across section. So cross section of a stem will have these components, a lot of components, but the components I want you to know are like this. Outermost part is epidermis, like anywhere, right? And then as you move inside, you go through different types of cortexes. The outer cortex, then you have the middle cortex, and then you have the inner cortex. It's kind of interesting. Why we have three types of cortexes is that they are the cells inside those cortexes are different. I don't want you to particularly remember or know the names of those cells. Like I do have them labeled on the left over here. I do have them labeled just for your information, right? On the outer and the inner side, those are parenchymatous cells. On the middle, those are sclerenchymatous cells, right? The difference is actually the thickness of the cells, right? That I do want you to know, that the cell thickness, the cell wall thickness is different, but the names of those sclerenchyma cells, I do not expect you to know, right? And then after you go through the three layers of the cortexes, right? And if I don't label them separately, if I draw a line, like a double-headed arrow, from the epidermis all the way to this particular structure, you can label them as cortex collectively. But if I particularly point out towards, middle, inner, or outer, right, then you will have to name them as middle, outer, or inner cortex, right? The innermost part is this vascular bundle. That actual part is called a vascular bundle, and the vascular bundle has two parts. One, you see large openings, right, with thick cell walls, right? Those are xylem, right? There in certain literature they label that as metaxylem, but for you just xylem is fine, right? So xylem, how do you know the xylem? Because it has larger openings inside and has a thicker cell wall. The other cells that are actually much smaller, right, are phloem. Xylem conducts water and mineral. Phloem conducts organic food, right? So xylem is largely dead, right? Those cells are dead, right? That's why, if they're alive, there will be resistance to water movement. Phloem cells are largely alive. Yeah, so you can actually label them yourselves on your own. So in the lab manual, we have a part about the fern leaflet. Just skip that. You don't have to do it. And then we are moving on to the last part. We are going through different displays. And again, this is a really cool part that I really love talking about. So here you are looking at the staghorn fern. It's a very beautiful horn. Why it is called staghorn, you should know by its appearance, right? Looks like a staghorn, right? Like horn, like it looks like horns. These branching leaves look like actually horns, right? And this part looks like the actual head, right? So if you can actually display, you can display this on a wall, right? And so it looks like a stag head. That's why it's called a stag horn. And it's an epiphyte. Can anyone tell me what an epiphyte is? I just explained that probably in the intro video, intro lecture.
Well, epi means outside.
Good, close. Epi actually means on the top. I think that's what you meant, Emily, right? It's been on the top. Phyte means plant. A plant that is growing on the top of another plant is an epiphyte. So this is an epiphyte. And they are not native to this particular area. You can grow them in this particular area, particularly in greenhouses. You can, and particularly in, there are people growing this actually in arboreta, but it's hard to grow them in colder environments because they are well developed for tropical environments. They are tropical rainforest epiphytes. And then how do they get nutrient is extremely important to know. I'm going to, so here, like I'm showing, this is how a single mass of plants looks like. It's, in this case, you actually, in this case, you see a single individual plant, right? In this case, you don't see a single individual plant, and over here you see a bunch of plants, same over here. So the reason is actually, in nature, always you actually have multiple plants emerging, right? From a single plant. So because they can vegetatively propagate and make a huge bush on the top of another tree, right? And they are huge structures, right? If you want to get a size perspective, I have a different photograph later on. So they are native to rainforests of, so largely Southeastern Asia and Australia, right? And actually in Southeastern Australia, where the temperature remains above 40 degrees year round, they grow really well. So in the United States, their growth, like in natural environment, happens much better in Florida, in the southern US, right? In our part of the country, we can't because we drop way below 40. Even in like southeastern US, like Carolinas, since the temperature go below 40, you cannot grow them outside. But in Florida, you can grow them. And I do suspect, I haven't read that, but I do suspect parts of the southern mid-southern part, like Texas, Louisiana, you should be able to get out with, you should be able to do something with these plants, because around the Mississippi River drainage basin, it remains quite warmer in those mid-southern states. So they actually have very short rhizomes, right? And they produce two types of fronds, right? The sterile frond, on the base, if you look at this particular part, that is the sterile frond, right? It's heart-shaped and it kind of is sort of overlapping, right? And it looks like a shield. Literally, the job of that sterile frond is protective. It shields the rhizome, right? It's quite dull green in color and then becomes more drier or browner later on. And it's earlier quite succulent and become more papery or more fragile as it matures, right? And of course, the older that frond becomes, the browner it turns. Its job, like I said, is to protect the rhizome and the root. It's extremely important. The protective function is very important because you're not growing in soil. So you have to have additional protection for the root structure. Otherwise, if the root dies, the entire plant dies. And also, the root, when they early develops, it actually gathers leaf litter and dirt from the atmosphere and creates a soil surface on the top of the epiphyte, top of the host plant, because it cannot actually collect any soil from the actual ground, right? So it has to create soil, right, by collecting decaying bark, decaying leaves, and any suspended solids on the atmosphere, right? So you have to keep that root from disintegrating. So this fertile front, this large fertile front, helps you do that. So the actual epiphytic soil is underneath this front. The fertile front then also forms brighter, greener, forked components like this, right? But if you look at the underside of that fertile front, right, you will see the sorai, right? And it's really interesting. The sorai occurs only on the tip of the fertile front, not throughout the fertile front, right? And they look like this. And if you get closer, they look like that, right? So you're looking at lots of sporangia collectively covering the entire underside, right? And being an epiphytes, they actually reside on the top of the canopy of another tree, so they have a really good chance of releasing the spores to the air. Right. And last part is about going over these different structures of sporangia, right? Arrangement of sporangia. First, strobilus. Right? Strobilus is sort of a corn-like part, that you find on the tip of the stem, and it appears like a corn, right? That's why, in fact, those club mosses looks like a miniature pine. And again, remember that there are small leaves, tiny leaves that sort of, sits below each sporangia. And then the second type of sporangia are sorai. Okay, and we explained that part quite a lot before, right? And those are found on the underside of leaves or underside of leaf blades. And again, this is additional photograph. I showed you this multiple times, right? And this is actually a good close-up how a single sorai look like. It has millions and millions of individual sporangia, right? And in fact, if you wonder what that is the annulus, right? That is the annulus. That actually has different cell wall thicknesses, and you can clearly see those differential cell wall thicknesses in that photograph, right? And then this one, we are looking at another type of sporangia where you have different fronts called fertile fronts that emerges upright, right, in the plant's body, right? And again, a close-up of what a fertile front looks like, right? And generally, the fertile fronts appear to be brown, but some cases, the fertile front also could be green, where they are not that different from the sterile front. Again, sterile front is vegetative. Their job is A, vegetative reproduction, B, photosynthesis. Fertile front's job is actually primarily to produce spores, so sexual reproduction, and or producing spores, and some of them are green, so they could photosynthesize for a shorter period of time. Sterile front could be observed year-round, right? Particularly, I mentioned that ferns are perennial, meaning that they are evergreen. So now again, there are exceptions to the rule. Okay, fertile sensitive fern, right, are actually not evergreen. They actually die after the first frost, right? So they are not evergreen, but most other ferns are evergreen, right? Most club mosses are also evergreen, right? So because of that, the sterile front has to remain active year round. But fertile front, which is only emerging when they're producing spores, may not be observed year round. They are actually seasonal, right? And most ferns only reproduce once a season. Yeah, well, that's it. That's it for the exercise. Now we can actually go through the homework. Do you guys have any questions? Alrighty, very good. Now I'm sharing the homework screen. Are we good? Give me a nod. You're looking at the homework screen. Alrighty. So the very first question, let's read it. Compare your notes and drawings of the sporophyte of mosses, liverworts, hornworts, ferns, whisk ferns, and ground pine. Now, ground pine, there are many names for those princess pines, right? Ground pine is one of them. In the space to the right of the each group, right? NV for non-vascular or SL for seedless vascular. List the characteristics of sporophyte in your drawings. In particular, A, note there, the column A, right? Note colors and sizes of sporophyte. B, location and the color of the sporangia. Column C, the presence or absence of stomata. D, the type dependency between the sporophyte and gametophyte. Now let's go and quickly discuss some of these columns. Column A first, where you are supposed to tell the color and the size of the sporophyte. Now remember, generally for all non-vascular plants, mosses, liverworts, and hornworts, all the time they are brown, right? Sometimes liverworts and hornworts, remember, they have green sporophytes, right? But most of them are brown, right? Mosses are always brown in terms of the sporophyte. And also remember that for all the non-vascular plants, the sporophyte is going to be always small, right? And nearly always brown. Okay. Now about the ferns. For all the ferns and club mosses or fern allies, right? Or fern relatives, always the sporophyte is actually green, right? Okay, now remember, I told you some exceptions. Forget that exception for now, right? They are going to be green in color, right? And they are also large. I know I provided you an exception, right? But generally for, like for general purposes, for all fern and fernalides and club mosses, green and large. Column B is about the location and the color of the sporangia. Now, color of the sporangia for all the non-vascular plants are generally brown, located in different locations, right? Remember, for mosses, remember they're actually on the top of the setae. Remember, it produces setae like a stalk, and on the top of the stalk, you have the sporangia. And then for liverworts, it's actually inside a capsule, right? If you remember, the capsule, right, are produced out of the female gametophyte, right? So, and then for hornworts, right, they're also located on that horn. Remember, it produces, the hornworts have like a horn-like structure on the top of the horn, right? It actually produces, it actually has the sporangia, right? And it splits longitudinally, if you remember. So the capsule part of the sporangia is located on the top of the thallus. It emerges from the thallus, sort of erects from the thallus. For ferns, if you remember, for the whisk ferns, right, we just covered them. Can anyone tell me the color of the sporangia?
Yellow.
Very good. It's yellow, right? And can you tell me where exactly it is found? You can say it is on the stem, but you can be a little bit more precise and say that either it's on the tip of the stem or at the forking point, branching points of the stem. Another way to say that is on the node of the stem, right? Node is actually where the leaf branching or branch formation happens. And in ground pine, can someone tell me the color? We just covered them, so you should remember. The color of that is what? Color of the sporangia? And the location maybe? So yeah, Emily?
Are they yellowish?
Yeah, they're yellowish brown, and where they are located? They are always located on the top, right, of the stem, right? Always on the sort of the pinnacle of the top of the stem. For true ferns, I did show you several types of sporangia, and they all brown in color, right? And the locations, I also showed you different type of locations, right? Some of them are as sori on the underside of fronds. Some of them actually produce separate fertile fronds, and some of them yeah, those two types, right? Either as a separate frond or as sori on the underside of the frond. And the C column is actually presence and absence of stomata. Now remember, out of all of these, which plant do not have any stomata at all? So easy to remember, all vascular plants actually, have true leaves. If they have true leaves, they should have stomata, right? That actually leaves the non-vascular plants, right? Some of those non-vascular plants actually have stomata in, well, all vascular plants have pores, right? All non-vascular plants have pores. Some non-vascular plants have stomata on the sporophyte, not on the gametophyte. But one vascular plant, non-vascular plant group has no stomata whatsoever. What was that unique group? Liverworts, right? Liverworts did not have, right? Everything else have everything else contain stomata, right? But I cannot emphasize more. Do know where you find stomata, right? Right? It's just knowing presence or absence is not good enough. Do know where they are located. D column, dependency. Now, think about dependency again, in terms of this evolutionary tree, right? When we were talking about non-vascular plants, right? I did mention that the sporophyte was completely dependent on the gametophyte, right? And I did mention that the sporophytes for non-vascular plants cannot effectively photosynthesize for itself. Even if the sporophyte appears green, they cannot photosynthesize for themselves. But when you come to ferns, club mosses, and fern allies, or fern relatives, always sporophyte and gametophytes are independent, right? Yes, when during the early developmental stages, sporophyte has to rely on the gametophyte for a very short period of time, but subsequently when it is mature, after it produces its first leaf, right, the sporophyte does not need to depend on the gametophyte. So for all vascular plants, the two generations of the life cycle are independent of each other. Moving on, the next question is, any clarifications regarding the table?
Should we mention that both can photosynthesize?
Yes, do mention. So for the column's sake, what I'm asking is whether they're dependent or independent, right? So you can simply write, sporophyte completely dependent on gametophyte, or sporophyte and sporophyte and gametophytes are completely independent. That is all I expect, but I explained the photosynthetic part so that you understand where that dependency come from. So the next, any other questions? Good. What trends do you see in the numbers of sporangia among these groups of plants, particularly between non-vascular and seedless vascular plants, but include all? What is an evolutionary advantage to number of sporangia and sporophyte? So let's talk, like, please do converse with me. This is actually a thought process. What do you guys think? What kind of a trend? First, that is a trend. Now, in terms of number of sporangia, what kind of trend do you see? And what is the advantage of that particular trend? That is a simple question. So let's talk. First, what trend do we observe?
Are there more sporangia with seedless vascular plants?
That is exactly correct, right? As you move up in the evolutionary tree from non-vascular to vascular, yes, the number of spores they produce becomes incredibly more, right? And how they do is quite different. To produce more spores, they produce more sporangia, right? This aggregation of sporangia in the sori, the production of different fronds, production of strobilus, all of those are efforts to increase the number of spores produced. All of those are efforts to concentrate spore production or energy and resources for spore production. Yes, so the general trend is increasing the number of spores produced, increasing the number of sporangia producing spores. Another thing that is important is actually, well, producing it is not enough. You need to have a mechanism to spread them better under the most ideal conditions and to spread them further away from the parental plant, right? So having specific ways to burst open the sporangia, having these lips, these lip cells, this annulum with different thicknesses, all of that actually helps them to actually enhance the success of spreading them, right? To piggyback on wind, right? To make them lighter, right? Those are kind of the trends that you see, the evolutionary trends that you see. Um, mm, Yeah, so that is actually basically the basic seed I wanted to give to you in your producing an answer for that particular question. Let's go to this particular table, compare and contrast phloem and xylem. Include a similarity. What do they look under the microscope? B, where they are located with respect to each other. C, their functions. So how do they look like? I mean, are they here? You can actually talk about for xylem. So the first column is what is unique to xylem. The last column is what is unique to phloem, right? The middle column is what's common to both, right? And A, what it based on the microscopic examination. B, the rows, where they are located with respect to each other. C, about their function, right? I mean, you know the different functions, right? I mentioned xylem transports mineral and water, both mineral and water, both. Phloem transports organic food, right? Complex organic food. And then another difference could be that for xylem transports actually mostly one way, right? From roots elsewhere, right? The reason I said mostly one way is that sometimes certain leaves have become succulent to store water. In that case, water can transport otherwise, from sort of reverse way, but that's a very, very exceptional situation. But mostly xylem are adapted to transport water against gravity, anti-gravity, right, from the roots upwards. So that's a difference. But phloem, it should be transported two directions, from photosynthetic organs to elsewhere, particularly to the storage organs, and then from storage organs like roots to elsewhere. So it has to, so phloem transportation goes along gravity as well as anti-gravity, right? So in terms of differences, the cell wall thickness, particularly xylem, since there's an extremely high pressure under which water and minerals are transported, the cell walls of xylem has to be very thick. That's why most xylem cells are actually dead, right? And in fact, a secondary function of xylem is to provide shape and rigor and stability to the plant body. You can consider the xylem as the skeletal equivalent of plants. But phloem does not have that rigidity. Their cell walls are actually thin. In fact, most phloem cells are alive, right? Only a few of them are dead. Well, similar device, they occur in a microscope, they occur as a single vascular bundle together, right? And they all help to move things, whether it's mineral, water, food. It's a conducting system, right? It's a transportation system, right? And again, if you compare the cell walls of any vascular system with the cortex cells or with the epidermal cells, you will see they're actually thicker, right? So compared to other parenchymal cells, right? Parenchyma is a kind of a plant cell that you find everywhere else other than vascular tissues and epidermis, right? Those parenchymal cells actually have the thinnest cell walls, right? Vascular tissues generally have relatively thicker cell walls. So those are some differences and similarities that you can include there. Oh, the next part, describe how true organs are different from structures with similar functions in non-vascular plants. So here we are comparing phyllids with leaves. We are comparing rhizoids with roots. You're comparing, yeah, that's kind of the comparison you're doing, right? So the difference is actually presence of vascular tissues, right? The true tissues have vascular tissues, both kind of vascular tissues, right? And then the next part, explain in general how a botanist can tell the difference between the three types of true organs. Well, that's actually something I want you to research on. What are the basic functions of the root? Absorption and anchoring. Stem, erection, upright erection, right? Exposing the leaves to maximum sunlight interception, right? Leaves, photosynthesis, right? So based on that, what kind of a structure would you expect histologically inside them? Where would you expect chlorophyll? Where would you expect absence of chlorophyll, right? Where would you expect cells that can actually store carbohydrates, right? So think like that, think about their function, and then try to relate that function to an anatomical feature that helps you do the identification. So for that, I'm going to ask you to do some research on your own. That's it. That is basically all I have for today. Again, like in all plant components, there's plenty of time to go over questions. Any questions you have regarding the homework that is due tonight, this homework, today's exercises, or your projects? I do not have anyone asking questions. Seems like we are all set. Anyone anyone who's actually losing interest about plants rather than animals? Yeah, I know. I get it. Remember, I've been in college. I was like you guys, and I regret that because I learned about plants way later in my life, actually, even after graduate school, like later in the graduate school and afterwards. That's when I fell in love with plants. Um, so you guys are not too late. You have a crazy plant crazy professor teaching you. So yeah, they're really cool. Well, guys, if you don't have any questions, we can call it a day, and I will stick around if there's anyone with other questions. If you do not have questions, I'm going to let you go.
Thank you.
Thank you very much. Have a great day.
I might
Everybody.
stick around a few minutes because I don't know if I have questions.
Of course, you can stick around in case someone else have questions. Okay, we have two, Monique and Emily. Monique, do you have any questions or are you hanging around in case someone does? Monique? That is strange. Yeah, we cannot hear you. So I don't think there are any questions, Emily. So I'll say goodbye to you.
Okay.
Are you sure you have any questions? Please feel free to ask.
I don't have any at the moment. I'm just concerned I might.
That's okay. Remember every time you have a whole week to ask questions. Right. I mean, you can ask questions next time or anytime else. So if you don't have questions right now, I'm going to let you go. Okay.
Okay.
Alrighty. Bye-bye.
Thank you.